Good morning, everyone, and welcome to our fourth Explained webinar series. Today we're going to be introducing the concept of medical research, what it is, and the different stages of medical research. Before we begin, as always, I'd like to thank our lovely sponsors, Camradis, who have funded this sixth webinar series. And remind you that we are recording the webinar, so if you miss anything at all, please don't worry, the recording will be made available tomorrow. So, our agenda this morning will be a first half-an-hour introduction from Oliver Timmis at the AKU Society about preclinical research, and then followed by Sarah from Raremark, who will be discussing clinical research. Then we'll have about half an hour for Q&As. So if you have any questions throughout any of the presentations, please do write them into your little questions tab, and we'll address them at appropriate question times. Okay, and all that's really left is to introduce our two speakers. So we did have Dr. Hannah Batchelor from the University of Birmingham, who is intending to give the first talk, but unfortunately she's feeling unwell, and we wish her a very speedy recovery. And so Oliver Timmis very kindly stepped in to give her a presentation in her place. So he is the CEO of AKU Society, which is a patient group helping those living with the rare disease alkaptonuria. And our second speaker, Sarah, is the lead community manager at Raremark, where she engages regularly with patient groups and the rare disease community to help them understand and be involved in the drug development process. And that's all from me. I will now hand over to Oliver to give us our first presentation.
Hi, so as Flora mentioned, I'm not Hannah. I'm Oliver Timmis from the AKU Society, and I wanted to give a quick introduction as to why I'm talking about preclinical research. So the AKU Society is a patient group, and we funded preclinical research in the disease alkaptonuria. So essentially what we found is that we had a potential treatment, a drug called nitisinone, but before we could do any clinical research, we had to do quite a large amount of preclinical research, basically showing that the drug works in models of disease. And the reason that we do that is that we can show that it's a safe and effective drug in a non-human model before we then do any human research. And the slides I'm giving today are actually from Hannah that she prepared for this presentation. So to start off, just a little bit of information about the drug development timeline. So essentially, it takes about 16 years on average to go from the discovery of a drug to actually having it as a available treatment for patients. And on the side here, you've got a brief timeline of how that works. So essentially, you have several years of preclinical testing, and then you go into the clinical testing, which I'm sure Sarah will mention later, but it's split into different phases, phase 1, phase 2, phase 3, before you then actually get to apply for a drug license. And one of the problems with preclinical testing is that it's quite variable. So if you look on the other side of the screen, that's the numbers of compounds or numbers of drugs in each stage. So typically in the discovery and preclinical testing stage, you've got up to 10,000 different drugs. And those are then narrowed down, so you start to see which ones are more effective, which ones are least effective, and then you start to narrow it down to the most effective possible drugs. As that goes through different stages, you end up eliminating the drugs that either don't work or don't work well enough, until eventually you end up with just a few going into clinical trials and eventually just one being approved by regulators. And the average cost of that is typically around $1 billion, which is an awful lot of money, and it shows that it's a very expensive, very time-consuming process. So what is preclinical development? So essentially, preclinical development, as I mentioned in the intro, is basically showing that the drug that you're testing is going to be effective. So basically, you're finding some hints, some clues as to the effectiveness of the drug. You're also looking to see if it's safe. So is it going to be causing another disease, or is it going to be making the disease that you're trying to treat worse? Is it stable, and is it excreted safely from the body? So that means it's eliminated through your urine or through other ways. And the regulatory guidelines for preclinical drug development is obviously quite very strict. And here are the two main bodies. So we have the FDA, the Food and Drug Administration in America, and we have the EMA, the European Medicines Agency for Europe. And essentially what they do is produce guidance about how preclinical development should work. And I'll go into some of the more details about the guidelines later. So these are the two major bodies, really. Obviously, there's bodies around the rest of the world, and there's also national bodies. So, for instance, in the UK, we have the MHRA, the Medicines Healthcare Regulatory Authority, which looks at just studies that are in the UK. However, of course, since the UK is still part of Europe, they also have to abide by the European Medicines Agency. And just a quick warning that I'm going to be talking about animal research in the talk today. And animal studies are actually a legal requirement for any new human drugs that are going to be approved in Europe. So essentially the reason for this is safety. It's to show if a potentially dangerous drug is safe to use in humans, we look to see whether it's safe to use in animals first. And actually the World Medical Association has in the ethical principles for medical research a requirement that new drugs use animal studies. So pretty much it's a worldwide standard to use animal research. However, one of the big issues with animal studies is that, of course, it's quite controversial, and researchers have to use ethical guidance to make sure that they use animals in an appropriate way. And one of the major principles, one of the international principles, is the 3Rs, which is reduce, refine, and replace. So essentially that's to reduce the number of animals to the absolute minimum that you need to use. It's to refine the processes that you use to make sure that you're not doing anything that's unneedly painful for the animals. And it's also to replace animals. So eventually, if there is a better way, a better model to use than an animal model, it's to replace using animals with a different model. And basic research is split actually into two, so preclinical research is split into two different types. So the first of those is basic research. So basic research, as it mentions, is quite basic. It's about discovering facts completely unrelated to any clinical aspects. So for instance, in 2006, the Nobel Prize for Medicine was won by these two researchers who showed that the RNA interference could be used to switch genes on and off. So that's a very, very fundamental piece of research that doesn't necessarily have any clinical outcome, but that's what's an example of basic research. Applied research is actually using that information in the area of treating and preventing illness. So that previous study about switching genes on and off can now be used in a clinical way to actually treat a disease. So if you've got a disease where a gene is expressed and that causes the disease, if you could then switch off that gene, then potentially you could have a treatment for that disease. So that's the difference between basic and applied preclinical research. However, before we even get to the preclinical research, we need to do preclinical research. So essentially what this is saying is that before you start doing any studies, you need to establish a target. So if you suffer from a disease yourself, you know that there's often a target for the disease. So there's a pathway that you're targeting, there's a specific enzyme you're targeting, or there's a specific tissue that you're targeting. And that's what researchers need to look for first. They need to look for a way to actually interact with the disease and have some kind of effect for its treatment. So preclinical studies. So I mentioned that animal research is a requirement for drug development. And so essentially what this is showing is that we need to, as drugs are going to be interfering with the human body, we need to show that they're safe before we start to use them in humans. And the way that generally is accepted is that we test out the drugs in an animal model first to show whether they are safe to then use in humans. So some important considerations. You need to look at the animal species and how many animals. So it may be that the best animal you can come up with is a massive animal like a cow, but obviously that's not very practical. So it's much easier if you use smaller animals, which is why things like mice and rats end up being used quite often. The other advantage with mice and rats is that they have a very high pregnancy rate. So you can have several generations of mice within a few years. Whereas if you used a different model, so a dog model or a cow model, obviously it's going to take much longer to have the same amount of generations. And the idea for animal research is that you can study a disease in a different model to humans in a much faster timeline. You also need to look then at the duration of studies. So you want to make sure that you're looking at an appropriate time. You don't want to end up spending 20 years looking at an animal model before even considering looking at human models, because obviously the aim of preclinical research is to get to clinical research as fast as possible. And we also need to look at the animal dosage compared to human dosage. So obviously, if you're going to be using a model such as a mouse, that's a very different problem than using a human. Mice are going to be much, much smaller. They're going to have much smaller bodies. They're going to have a much higher turnover metabolically. And so the dose that you need to use in a mouse is going to be very different to the dose you use in a human. And so you need to take that into account when doing any research. And the question at the bottom is, what level of evidence do you think is required to allow a new drug to be tested in humans? And essentially what we need to show is that we're absolutely certain that the drug won't cause undue harm to a human patient. So essentially what we need to look at is the risk-benefit. So we need to show that the benefit of the drug is going to be better than the risk of it causing damage to the patient. So one of the first things we look at is toxicology studies. So essentially this is looking to see whether the drug is safe. So toxic effects could be, for instance, on the slide, there could be effects on reproduction. So it could be that a drug is making it impossible, very difficult to reproduce. It could cause things like cancer. It could cause things like genotoxicity. So that's when you have problems with DNA changes due to a new drug. It could have an effect on the immune system. So obviously some drugs target the immune system. So if you're looking, for instance, at a drug to combat an organ replacement, you actually want to reduce the effects of the immune system so that the body won't reject a new replacement tissue. And then potential to cause skin and eye problems. So essentially what toxicology studies look at is the broad range of side effects. And what you're trying to aim for is the minimal side effects possible. And moving on is dose ranging and dose finding studies. So obviously once you've got an idea of a drug that potentially could work in a disease, you need to look at the actual dose that the drug is going to be used at. And essentially this is how researchers will look at it. So if you look at the graph up in the top corner, you've got the amount of drug on the bottom axis, and then you've got the effect on the body on the y-axis. And typically, as you'd expect, if you increase the amount of drug, you're going to increase the effect on the body. And this is where you need to get to a bit of a balancing act, because if by increasing the amount of drug, you also increase the side effects of the drug, you end up reaching a threshold where you can have the maximum benefit from the drug and the minimal side effects from the drug. And that's what is one of the things that we look at in animal models, is looking at the different dosages and see how they relate to effects in the body. And the issues in toxicology studies. So how do you measure the dose effect on toxicology? So I mentioned on the previous slide that we typically assume that increased dose means increased toxicity, but is that actually true? There could be something else happening, and it could be that actually by increasing the dose you reduce the side effects. So that's one of the things that we need to look at in animal models. And the other problem is that we're testing drugs in animals such as mouse. And one of the advantages of using drugs in humans is that you can actually ask them about the effects. So if you test a drug in a human, you can ask them, are you feeling pain? Are you feeling sick? Are you feeling dizzy by taking this drug? You can't do that in animals. So it makes actually looking for some of the side effects in animals more difficult, which is why sometimes when we get to the clinical research stage later, that new side effects are discovered because they're only side effects that can be communicated by a human to their doctor. So the first thing we look at is the short-term toxicity. So typically animals will be observed for 30 days. So for instance, if we're going to be using a mouse model to test a new drug, there'll be a certain number of mouse that's decided by the statistical review before we start the study. So say that we have 100 mice, they're each taking the drug. We can observe them for a month, and then we can look at their different habits. So the researchers that do any mouse experiments are very close often to their mouse colony, so they know what they're typically like when they're healthy animals. And so they can see if there's any changes to their typical habits. So if an animal starts eating or drinking less, or eating or drinking more, they can pick up on that. One of the obvious changes is a weight change. So typically in animal studies, the mouse will be weighed regularly, perhaps every day, and then they can look at changes over time. So if the drug is causing an increase in weight or a decrease in weight, that could be monitored. We look for toxic effects, so that could be things like the quality of their skin, the quality of their fur, it could be lesions in their eyes, it could be any number of effects that are obvious to see. and also psychomotor changes. So, for instance, if a mouse no longer can walk straight, or if it seems to be dizzy or losing its balance, that's also something that can be observed. So typically in these studies, the mice are actually very well looked after, and they're studied very, very intensely because we're looking for exactly these kinds of changes. And the next is quite slightly longer term studies. So these are typically over longer days. So they're normally 90 to 180 days, so that's 3 to 6 months. And what we're looking for is the different changes over longer time. So we typically use human administration at this point. So in the shorter term studies, the drug could be put into something like the mouse is drinking water, so they take the drug that way. In these experiments, typically it's done in the same way that it's administered in humans. So that could be via injection, via an inhaler, or other ways. And what you're looking for in the mouse model is a longer term study than in humans. So for instance, for a chronic human disease, so a disease that happens over the lifetime of a patient, you'd be looking to do about a year of animal testing. And one of the things that you're looking for by doing all this research is the no observable adverse effect level, or the NOAL. So essentially what this shows is the highest amount of drug for the lowest amount of side effects. And that's exactly what you want to find in a dose ranging study. So you want to know what dose is going to be the best dose to then use equivalently in humans. And this is some example information from a toxicology study. So I think Hannah prepared the slide, but this is going to be from a study report. And essentially what this shows is the study shows that holes are forming in the kidney and in the bladder of mouse. And sorry, in rats, and obviously this is very important information to find out about a drug. Obviously, this drug has quite serious side effects, but it's the kind of thing that you wouldn't be able to do in humans. So if you did a human study, for example, in this potential drug, and you found out that you were causing this level of damage to a patient, that would be completely unethical. So that's why we tend to do research in animal models before we move on to human models. And one of the most important things to look for is carcinogenicity studies. So this is essentially showing, is the drug causing cancer? So for that, we tend to use different types of rat and mouse. So these are rats and mice that are much more susceptible to developing cancerous tumors. And we can use those to see if there's a slight chance that the drug causes cancer, because obviously in these mice, they're already more susceptible to developing tumors. So if there's any way that the drug can lead to cancer or tumor formation, then these mice are going to be showing it. We use doses similar to a human study so that we can see the effect that it would potentially have if the drug was used in human patients. Reproductive studies, so I mentioned earlier that one of the main things to look for in toxicology studies is the effect on reproduction. So this looks on effects on fertility and mating. So essentially, I mentioned earlier that mice have a much faster turnover. So mice typically tend to live for 3 to 4 years, and they'll have new generations of mice every couple of years probably. And so obviously it's easier to then study the effect on reproduction in that kind of model where you don't have to wait for 25 years like you would in a human model. Safety pharmacology. So the next thing to look for is how the drug interacts with the body. So we know that how the drug metabolizes. I'll show you on the next slide a quick diagram of that. But this shows how the drug can affect vital functions. So some of the typical ways that drugs will interact with the body is via the kidneys, because obviously the kidneys will produce urine, and all drugs end up leaving the body through the urine. Through the GI tract, so especially if you're taking a tablet that you're swallowing, to the cardiovascular system, to the blood, as that's the main way that drugs circulate through the body, and then through other body systems as well. However, there are some problems here. So, for instance, in rat models of diseases, rat kidneys are actually much more efficient than human kidneys. So, essentially, rats produce a lot more urine than humans do, and so the blood of a rat goes through the kidneys much faster than the blood of a human goes through human kidneys. And so there are problems with animal models, and that's one of the examples. And it's one of the things you have to take into account when using a non-human model to study a disease. So I mentioned about we know how the drug interacts with the body. So typically, if you're taking a tablet, you'll swallow a tablet that will then go through your esophagus into your stomach. It'll then go through your intestines. The drug will then usually be absorbed through the intestine wall into the blood. It will go to the liver straight away, and the liver will start to break down drugs. That will then go into the systemic circulation, so into the blood, and then will circulate around the entire body. Some of that will go into the kidneys and will be excreted in your urine, and some of it will actually reach the tissues that it needs to go to. So for example, if you take a painkiller like aspirin, so say you've got a pain in your foot, you swallow a pill, that then will go into your stomach. Aspirin actually gets absorbed across the stomach lining. That will then go into your liver, that will then go into your blood. The blood will then take it down to the foot, and then the aspirin will actually reach the tissue that's in pain, and then will have an effect. One of the things to mention here is that at every stage, you're going to be losing medicine. So not all of the medicine will be absorbed across the intestine wall. Some of it will go straight out and be excreted as feces. Some of the medicine will cross the intestine wall and go to the liver, and at that point, some of the medicine will be broken down by the liver. So that's the function of the liver, is to break down toxins in the body. So it will break down some of the drug. Once it enters the blood, some of the drug will then go to your kidneys and will be excreted as urine. So every stage of this process, you're going to be losing medicine. And it's one of the big reasons that we have to do dose studies, because it's not always obvious how much drug is going to get to the target tissue. It's definitely not going to be 100%, we know that, but we don't know for certain how much will actually reach there. So that's why dose ranging is very important. So the typical outcome from preclinical testing. So this is from an SMPC, so that's a summary of product characteristics. And this is an official document that has to be submitted to the European Medicines Agency when applying for either a drug license or applying for advice about doing clinical studies. And essentially what the summary shows is what you've found out from the preclinical testing. So as you can see in that first paragraph, it shows that there's no special hazard for humans based on conventional studies. So that's what you're looking for, really. What you're looking for in all of this animal work and in all of the preclinical work is showing that you're not going to be causing unnecessary harm to humans. And once that you're fully convinced from all of the animal work that you're not going to be causing undue harm, then you can start to consider doing clinical research. So biomarkers are essentially surrogates for problems in the human body. So you may have a disease, and that disease may show by an increase in a certain chemical within your body. And quite often, preclinical studies will look to identify that specific chemical, that biomarker. And ideally, that chemical will be very specific. It'll be something that's really obvious to see in a lab. And it will also be, you'll be able to look at it non-invasively. So you won't have to do surgery or anything to find the chemical. You'll be able to do it via an X-ray or some other kind of imaging technique. Obviously, the advantage of that is that you can look at the effects of a drug very easily without sort of any problem to the patient. The success in preclinical studies, so why are you actually doing this? So essentially what you're looking for in preclinical studies is that the drug is effective, it's safe, it's stable, and it's excreted safely from the body. So that's exactly what you're looking for in preclinical studies. And if you can show that, you're determined to be successful in your work. And obviously you have to get agreement from regulators before you then consider doing any human work. I mentioned before that animals, such as a mouse, has a very different body type and body size to a human. So you need to look at the dose that you're going to use. I'm not going to go through the calculation, but essentially, if you look at the very final line, the dose given to humans are about 6% of the dose given to mice. So essentially what this is showing is that the dose that you use in a mouse is much, much higher than any of the doses that you'll use in a human. And essentially that's to show that if you can show that the drug is safe at a much higher dosage, you can be confident that the dose will be safe to use in humans. So just to summarize, so preclinical studies that are undertaken to ensure that medicines are safe and effective before you get to humans. It takes a long time and costs a lot of money. So I showed earlier that the average cost for the development of a drug from start to end is about $1 billion. Quite a large amount of that is in preclinical studies, often because you're looking at several different drugs at the same time. And there's many people involved to ensure that the first clinical trials are safe. So there's no way that we can do clinical trials in humans without being fully confident that you're not going to cause unnecessary harm to a human. And so a lot of the preclinical work is just showing that you're going to be confident that you won't harm humans. Thank you very much for listening. If you've got any questions, I'll take it later. And I just wanted to share one extra link, which Hannah included in the slides, and that's to the ABPI, the Association of the British Pharmaceutical Industry website, and they have a schools website, which includes very good sort of basic information about all stages of drug development, including preclinical and clinical research. Thank you very much for listening, and I'll take questions later. Thanks.
Thank you very much, Oliver. Before we move on to Sarah, just because we still have a minute or so, just one question that's come through that I thought I'd hand over to Oliver, which was, you mentioned that reproduction is a important thing, and the question that's come through is other animals like rabbits also mate very regularly, so why are mice and rats used more than rabbits, for example?
Yeah, good question. So, I mean, there are examples of pretty much every animal being used as some kind of disease model. So there are research done in rabbits, but typically mice are used just for practicality. So they're very small, they breed very fast, and they're very cheap to look after. So kind of in terms of food and drink and heating and accommodation, they're very cheap to use. So typically mice tend to be used as a standard for it. One of the other advantages of using mice is that we know their genetics incredibly well. So mice have sort of become the standard for preclinical research. And so that means that a lot of research has been done looking at the genetics and kind of the causes of disease in mice models. And so often it's easier to then do subsequent work in mice just because we know so much about them already.
Brilliant. Thank you very much, Oliver. If there are any other questions, do just send them through. But for now, I will hand over to Sarah to give our second presentation.
So thank you, Flora, for the introduction, and Oliver for that presentation on preclinical research. I'm Sarah. I'm the lead community manager here at Reproduction. I'm a biochemist by background, but for the last 5 years have worked in the pharmaceutical industry, helping pharma to really understand patients and their needs, and also to help families and patients understand research. So I write educational content here at Remark about research and clinical trials, and we also connect families affected by rare disease to research opportunities. So hopefully in the next 30 minutes or so, I can give you an overview of clinical research and the steps, and also do a bit of jargon busting as well, knowing that if some of you are looking at trials online or looking at trial protocols, so an overview of a trial, there might be some phrases on there that are commonly used that I can go through. I almost certainly won't have covered everything, so if you do have any questions, I'm happy to take them at the end. And if I don't know the answer, I've got a whole team here that I can go back to, who've been working in drug development for 20 plus years, so we'll definitely get back to you on that. So in this part, I will cover what clinical research is, who the key players are, why people might take part in clinical trials, what the stages are, and then, as I said, hopefully explain some terms that you might commonly see being used when it comes to trials. So what is clinical research? You may also hear it being referred to as some of these phrases on the left side here, so clinical trials, clinical studies, human trials. So when we talk about clinical research as opposed to preclinical, we're generally talking about studies with human participants, whether that be healthy volunteers or people with the condition themselves. So just the definition here, clinical trials are conducted to collect data about the safety and effectiveness of a potential new test, drug or device before it is approved or widely used, and it's in people. So who are the key players in clinical research? Well, first you have a sponsor. So this is the sponsor of the research, and essentially they are funding the trial. So typically this is a pharmaceutical company or a university, but it could also be a patient group or charity, or involve more than one of these groups, so a pharma company or a charity in partnership who are co-funding a trial. It could even be a hospital or a single doctor who perhaps wants to look at a specific drug that's already approved in a subset of patients in a kind of single trial. Ethics review boards are also used. These are independent groups responsible for the protection of the rights, safety, and well-being of people taking part in a trial. They have to approve a clinical trials protocol, which is the document detailing the trial design and how it will be conducted before it can go ahead. The principal investigator is the person who leads the research team running the trial. They're usually a specialist doctor or researcher. There's also a study coordinator who supports the principal investigator. So this person is in charge of the day-to-day running of the trial. They help to coordinate activities between the principal investigator, so the key doctor, the sponsor, and the ethics review boards. And obviously for much larger clinical trials, there may be multiple study coordinators working together. And last, but certainly not least, there is the participant, which is the patient or the healthy volunteer taking part. So these are the key players, but there will also likely be multiple doctors and nurses across hospitals in many different countries helping to run and coordinate all different aspects of the trial. So why do people take part in clinical trials? Well, research is really the only way to speed up the approval process for new drugs. So using something off-label, which is essentially using something in a particular condition that hasn't been approved yet, but is shown to work anecdotally, or a doctor or has seen it work before. It will only lead to it actually being approved in that condition if research is then conducted into that condition. So trials help speed up the approval process. Some people take part in trials so they can see a specialist in that condition for the duration of the research. So particularly in rare disease, where it might be very difficult to be seeing regularly a specialist into the condition. Often it's these specialists that are conducting the trials in a site further away or in the same country. And so taking part in a trial often gives access to this specialist care. It's also potentially the only way to access experimental treatment that might be being used in the country. And the nature of trials means that you are more likely to be monitored more closely during that time. I wanted to quickly go over some of the different types of trials you might see, especially in rare conditions. So diagnostic trials study new tests or procedures for diagnosing diseases. So that's pretty self-explanatory. Natural history studies look at how diseases might progress naturally over time. So this is something that's often done in rare disease to look for aspects of a condition that the researchers can target to develop treatments for. So often you might see this in genetic diseases of researchers following somebody from the point of diagnosis without any intervention at all to understand more about how the disease progresses naturally. Observational studies monitor participants without intervening at all. So a natural history study is a type of observational study, but the person being monitored is not on any drug or intervention. It's the natural progression. Whereas an observational study, the person taking part might be on a drug already, or perhaps have had some surgery that they want to monitor afterwards without intervening during the trial, but there is still an aspect to the trial in terms of observing. So they might already be on a treatment, or they might have just had a specific surgery. Screening trials test the best way to detect certain diseases or medical conditions. So trials like this often lead to new ways of identifying conditions, or so for example, if a disease has a particular symptom, such as kidney disease, they might do a screening trial to look for that genetic condition in all of their patients with kidney disease. And finally, what you might be most familiar with and what we'd ideally like to get to in every condition are treatment trials. which evaluate the effectiveness and safety of potential new treatments. So clinical trials are split into 4 phases or stages, and I'm going to give a basic overview here of what each phase is about. So phase 1, this is typically a small study with anything from 5 to 100 healthy volunteers, depending on the nature of the condition. So phase 1 studies assess safety. So once you've gone from the preclinical research and you have identified that it's safe and effective in mice models, as Oliver was saying, it then moves into phase 1 at a very low dose, and it's given to healthy volunteers just to check the safety of the drug being tested. In rare disease, sometimes the drug is actually given to people with the condition, so you could think of it as maybe skipping phase 1 and not healthy volunteers as a safety study. This is so that the drug, if it works, it can be identified and approved faster and really be tested with people who actually need it. So phase 2, once the drug is shown to be safe, it moves into phase 2. So this is normally a larger study with people with the condition, and this could use a placebo. I'll explain more on placebos later, but they are essentially a drug, they contain no active ingredient, and it's normally given to some people in the study to act as a comparison. Phase 2 studies typically look at both safety and effectiveness. So phase 3 are larger studies still, and they're normally randomized and controlled. And again, I have more on that jargon busting later. But this is really a trial that leads to approval. So again, in rare disease, sometimes phase 2 and phase 3 are combined to speed up the process so that the drug can get to the approval stages quicker if it's shown to be effective. And it's at this point in phase 3 that typically researchers will try to look at dosing. So they might increase the dose as the trial goes on to understand what's really going to be as effective as possible while still being safe for the people taking part. So finally, if the drug has been approved after phase 3, so phase 3 is the trial that leads to approval if it's shown to be safe and effective, the drug actually still is continued to be monitored, and these are known as phase 4 studies. So the drug is approved for use in particular countries, but the researchers continue to monitor the long-term safety and effectiveness in the real world. And often this is really where you can see what impact the drug is having on the people that need it the most. And these are known as phase 4 trials. Just to give you an idea of how many drugs are typically making it through from preclinical to phase 1 and beyond, about 70% of the drugs that go from preclinical to phase 1 will then make it into phase 2. And of that, about a third of the drugs that have gone into phase 2 will typically be shown to be safe enough to move it into, safe and effective enough to move into phase 3. And then by phase 3, around 70 to 90% of those drugs will hopefully make it to phase 4, so they'll hopefully make it to approval. So these are just rough estimates, but it does give you an idea of the nature of the difficulty of drug development, I guess. And as you know, in this clinical research stage, this can take anything from 6 to 10 years plus. And then obviously when you add preclinical, that's when you start to get into 16 years plus, as Oliver said. So what happens during a trial? This is a really brief overview of the steps, just so we can understand what's actually happening at each stage. So as I mentioned before, the sponsor, who could be the pharma company or the doctor or a patient organization, designs the trials with the help of the doctor or the principal investigator. And they'll give an overview of this trial as a trial protocol. The doctors or investigators then see patients and they collect data from them, such as blood tests, observation, their own well-being in their own words. So you might see this as quality of life from the patient's perspective. And they'll enter all of this data into a database. It's then analyzed and presented in a report. And then, if it's phase 3, the report is then used to write an approval application for the regulatory bodies. So I believe Oliver mentioned two of these before, the FDA in the US and the EMA in the EU. I have more on those as well later, but these are the two regulatory bodies that these approval applications are being submitted to once a drug is shown to be safe and effective. So in general, a clinical trial might actually look like a few extra normal trips to the hospital to see a new doctor. So you may find you're going more than normal or having more tests than usual, and obviously you're receiving potentially an experimental drug. They're also increasingly happening at home to make it an easier experience for the patient, because as we all know, clinical trials aren't the easiest thing to do, and they're also not the easiest thing to attend, especially if you need to go frequently to a hospital that may be far away for you. And in larger trials, so phase 2, phase 3, the trials are often randomized, double-blind, and placebo-controlled. So I wanted to explain this term in this session, as it will likely be the most common words or phrases you see when you're looking for trials online or thinking about taking part in trials, whether that's rare disease or more common conditions. So randomized, double-blind, placebo-controlled trial is a common way to design a trial, as it ensures that the data that comes out of that trial is robust, reliable, and has reduced the bias as much as possible. And this is almost one of the standard designs of the trial that will then lead to approval, especially if no current treatment exists in that disease. So what does this actually mean? So I'm going to break down this phrase into its individual parts. Placebo-controlled. So placebo contains no active ingredient and is given to some of the participants so that the effectiveness of the drug can accurately be measured. So this could be something like a sugar pill. The participants are then randomly assigned to either take the active drug or the placebo. So then, to reduce the bias and not influence anyone, both researchers and patients don't know who is on the drug and who isn't. So they don't know who's receiving a placebo versus the actual experimental treatment. So this type of study is known as double-blind. And all of these aspects together allow the study design to be as reliable as possible when it comes to analysing the results. So not all of the studies that you come across will be placebo-controlled. As I said, this often happens if there's no currently available drug, just to make sure that to reduce the impact of the placebo effect, so people feeling like they're getting better. If there is an existing trial, sometimes it's more ethical or more reliable even to have the control group to be on the current standard of care, so the current treatment, and have that group then compared to the experimental treatment. Because often all you'll need to be identifying is if the new drug is giving better outcomes than the drug that's currently available. So what happens after a trial? If the drug is shown to be safe and effective in trials, then the results are submitted to a regulatory body such as the US FDA, the Food and Drug Administration, or Europe's European Medicines Agency. And they decide if the drug is effective enough to be approved. So this process, as you can imagine, takes a very long time. But in a nutshell, the data is reviewed by the experts there to ensure it's reliable. And they'll look at things like the impact of, is it making patients more mobile? Is it allowing people to go back to work? What is the health economic impact of the drug? Is it going to reduce hospitalizations? So even if it's expensive, if it has cost effectiveness as well as being safe and effective, then these are all the elements they're looking for to consider an approval. So once they've reviewed this, they might even go back to the sponsor and say, we need additional data in these areas, or we need you to review this particular aspect of the research you did before we can go ahead. And the sponsor will then go away and be expected to run additional trials to achieve the approval. Or if they're happy with the results, they will approve the drug, and the drug will enter into phase 4 trials, as I mentioned before, where it's continued to be monitored in the real world. And in terms of publication of the results, well, Not all sponsors, unfortunately, currently publish the results of their trials. However, there is a movement towards making the research more accessible to people. So hopefully the results of trials are then published onto PubMed, which is a medical research database, which you can go online now. It's free to access to search any research at all using keywords. Or they are published to clinicaltrials.gov, which is the US National Institutes of Health website, where it's really the biggest or the most comprehensive listing of clinical trials going on around the world. It is very US focused, but if you do want to go on that to look at research or even to look at trials that are happening at the moment in a specific condition, you can, and you can search by which ones are industry sponsored versus university sponsored, which ones are recruiting, and hopefully there are contact details on there as well. So I'd recommend some of those resources if you're looking for clinical research or published clinical research. But as I said, it's not a requirement to publish either trials or research on either of these sites. But hopefully there is a movement to doing that more frequently for all the sponsors. So I hope that's given you an idea of what the research process looks like. All this information and more is available on the Raremark website and through our newsletter. We also do a lot of work in clinical trials for children, which I know I haven't covered in this session, but there's a lot of our blogs and educational materials cover that. So please do let me know if you would like to receive any of this content by emailing me. We'll also be sharing it widely in May for Clinical Trials Awareness Week. And I hope that's given a good overview of clinical research. So if there are any questions, please do pass them on and I'll be more than happy to answer them.
Thank you very much, Sarah. I think we've already had one question through, so I'll just ask that for you immediately. We've had a question to just clarify whether the rules about clinical trials are standard across the world or if they're unique to Europe and different regions. So it's not just about who's approving, but just generally what has to be done within a trial. Is that standard or is that unique?
Yeah, sure. So it's a tricky question because the ethical guidelines generally, or what can be done in a trial, is standard in terms of the people working in the industry know what's required of them, and as much as possible, the ethics involved in research is pretty standard. However, in terms of official documentation, it does actually vary from different countries, even across different ethics review boards. So when I talked previously about ethics review boards, they are essentially a group of researchers, lay people, patients, people completely unrelated to the pharmaceutical industry at all, who get together perhaps once a week, once a month, in various universities, or it could be across something like the NHS, and look at thousands of applications for research and decide whether they're ethical or not. So sometimes the guidelines or the rules can even come down to what that ethics committee finds to be acceptable. So in terms of designing a trial or understanding what's ethical for patients, you'd hope that people all have the right ethics in mind and the right guidelines in mind. But in general, you will be looking for the guidelines to your own regulatory body. So in the US, that's the FDA, and in Europe, that's the EMA, and also to make sure that you have the right ethics and guidelines in place for the local or national ethics committee that you'll be submitting your research to.
Brilliant. Thank you, Sarah. Another question we've had through, and please, everyone listening, if there are any points for clarification or you want to delve into anything, just type it into the questions dashboard and I'll ask them on your behalf. So one question we received for Oliver is about the ethics around using animals in research, and what is the process that you have to go through? Is there an ethical process you have to go through to be allowed to do animal research?
Yeah, it's a good question. So as I mentioned in my presentation, animal testing is a requirement for any testing of drugs before you look at humans. And so obviously it's quite a widespread practice, but it is, as you say, sort of quite controversial, and there has to be ethics in place to make sure that what you're doing to the animals is correct and in no way inappropriate. So in the UK and in most other countries, the researcher has to apply for a license. So in the UK, you apply to the Home Office and you get a license to be eligible to do animal research. So you have to prove that you have scientific background, but also you have to prove that you understand the needs of the animals. And then for every specific study you do, you have to apply separately to an ethics board, explaining exactly what you're going to do to the animals. So how you're going to administer the drug, how you're going to look for side effects, and what you expect will happen to the mouse or to the animal model. And that ethics committee then has to approve for every different study exactly. Have to approve to give you permission to do that. And if you make any changes to the study, so for instance, if you previously said that you're going to study just the back right leg of the mouse, but then halfway through the study, 6 months later, you realize actually it'd be quite good if you could look at both back legs of the mouse because it would be a comparison. That's a change, so you then have to go back to the ethics committee and ask permission to do that different study. It's a different study in their eyes. So it is very, very heavily regulated. You can't do anything without any kind of ethics permission from an ethics board. And those ethics boards typically comprise of researchers that specifically study ethics. They come from experts in the disease, and usually they have a patient and public representative as well. Yeah, you can rest assured that any animal research done in the UK and in most countries will be very heavily regulated to make sure that it's appropriate to the mice.
Brilliant. And as always, when talking about trials and Brexit, there's a Brexit question. So I'll hand it first to Sarah and then Oliver if you have anything to add. With Brexit happening, what do you think will be the effect on research and monitoring?
So I think the short answer is I don't think we really know. I imagine that with Brexit happening, we obviously there will be, as far as approval is concerned, there will need to be approval from the EMA, but then I believe that we will also need to have separate approval systems for the UK alone. So I'm not entirely sure what's going to happen from my end. I'm probably just watching it closely. I don't know if you wanted to add anything, Oliver.
Thanks, Sarah. Yeah, so similar, I guess. We don't know for certain what the effect will be. All I can say is that most of the regulations that the UK use are regulations that were developed by the European Medicines Agency, so they're European regulations. But saying that, the UK quite often had a big voice in making those regulations, and there's bodies such as the MHRA, the Medicines Healthcare Regulatory Authority, which is just a UK body that does regulation for research. And I think to the most extent they will follow the European guidance that's already there. So I think probably the actual guidance won't change significantly. There may be minor changes here and there, but I don't think that's going to change. The big impact that I think we're going to see is a reduction in funding. So from the AKE Society, we actually are doing clinical trials at the moment called Develop a Cure, and those are funded in full from a European Commission grant. And I think the ability to no longer apply for those grants is going to have a pretty serious impact in clinical research in the UK.
Yes, this is something just from personal experience to add to that anytime I go to conferences, either in rare diseases or generally, this question gets asked, and pretty much the answer currently is we don't know. And I think it's going to be a very key aspect in the Brexit discussions and the changes in laws and regulations that we really need to push as the medical research society that we're part of to make sure that the best possible rules and regulations are enforced following Brexit. So the best care is given to patients and the most appropriate rules are in place. So another question we've had through are, I think towards Sarah, is what are the best ways to identify the key players in clinical trials and to approach them as a patient group to get involved?
So I think the place to start with identifying who the key players are would be clinicaltrials.gov or any publicly available information that you can find on the trial. hopefully the best case scenario is the sponsor has gone as far as to create a website specifically for the trial, but at the very least has listed their trial on clinicaltrials.gov. And there's a kind of spectrum of quality of information there, but you'll at least be able to, at very least, find the sponsor names, the name of the pharmaceutical company. And from there, you may even been given a contact information or locations of the trial sites. So that at the location of the trial sites, they'll usually list the principal investigator, so the key doctor. But in terms of being a patient group and approaching the key players, I would probably advise to approach the sponsor themselves and to try to find the contact information of the patient advocacy lead at the sponsoring the pharmaceutical company, or the key doctor there, or the chief medical officer. So as a patient group, they'll definitely be wanting to hear from you. And so I would go straight in at the top almost to the sponsors of the research, to which hopefully the information is either listed on their website or on clinicaltrials.gov.
Brilliant, thank you, Sarah. And now a question for Oliver. As patient groups, what can be done to encourage basic research to make sure that the proper foundations are in place before something goes to clinical trial?
That's a great question. I mentioned I run the AKU Society, which is a patient group, and we were very much involved with the clinical research before we started doing any clinical trials. And we actually supported it financially. So our model essentially was to look at supporting PhD students. So we were lucky, really, that there was a couple of labs that were already doing some basic research into alkaptonuria. But the way that we found that we could really influence it was by supporting specific students doing their PhD to then focus on a specific topic. So we could kind of then push the research into areas that we thought was going to be the most appropriate for the disease. Obviously, PhD students, they will spend maybe 4 or 5 years studying a specific area of a specific disease. And so that's quite a valuable way of doing it. And to be honest, it wasn't a particularly expensive way of doing it. So if you sponsor a PhD student, that's roughly 30 to 40,000 a year for 4 or 5 years. So it's something that is still expensive, but something that is perhaps within the scope of a patient group to actually support. A lot of the other research, so some of the research was much more expensive. So for instance, the development of an AKU mouse model, that was a research project that probably costed about £500,000. That was sponsored through us, but through a grant from the Big Lottery Foundation. So back in 2008, the Big Lottery Foundation offered research grants for exactly this kind of research. And so we worked with research teams to put together an application to submit to the lottery, and we supported it as the patient group, and we were lucky enough to actually get funding to then support development of that. So I think definitely that kind of research support role is definitely something that patient groups can do. The other thing that actually is very beneficial for basic research is things like human samples. So AKU, for instance, is a disease where patients end up needing joint replacement operations. So whenever a patient goes in to have a replacement hip, for example, the old bone is cut out of the body and a metal joint is put in. That old bone can actually be donated to medical research. And for preclinical research, that's really, really a vital thing. So most patients in the UK with alkaptonuria, they'll contact us if they're going to be having a scheduled surgery. We then put them in touch with the research teams to then donate that sample, which then becomes a very valuable resource for them. And I imagine in most diseases there's going to be something similar. Even if it's something like blood and urine samples, that's still really important resources for basic research.
Brilliant, thank you, Oliver. So while we wait for some more questions to filter in, please do keep submitting questions. I just wanted to let you all know of some upcoming things happening at Find a Cure that you could all get involved in. So we have a workshop coming up which will be looking at community fundraising, which will be at the end of April in London, and there's the link to sign up. This is free to attend for any patient group, and we will help patient groups with travel expenses as well to help make sure that you can attend regardless of where you're coming from within the UK. We are also going to be holding an event in Cardiff in early May, on May the 4th, and that will be a networking event, so that won't just be for patient groups, it'll be for anyone involved in rare disease research, really to highlight some of the excellent research being done in Cardiff and in around Wales. Again, that's free to attend for patient groups. There is a very small fee for researchers and pharmaceutical companies. And then finally, and a small plug for our online portal, we will be having a very short webinar just to introduce some of the functionalities of our portal. For those of you who aren't aware of this, this is a central resource hub that we've created with information specifically written for rare disease patient groups around how you set up. There's quite a lot on there on medical research and clinical trials and working with academics and working with pharma, so you might find it useful to look on there. And so this webinar will be introducing different ways to use the portal that you might not be aware of just up front. So I'll just leave those up for you. And I believe we've got a couple more questions. So yes, just one question, I think, towards Oliver. So some funding organizations are opposed to giving funding to any charity that supports animal research. What are your thoughts on how you communicate around this, or how you still get funding in these situations?
Yeah, good question. So I mean, it goes back to the what I was saying earlier, that animal research is quite controversial, I suppose. But as I mentioned, in Europe at least, the European Medicines Agency has a requirement that any new drug is tested in animal research. So it is a reality of doing any kind of drug development that at some point it will need to go into an animal model. So there are funders that don't support charities that support animal research. I've come across a few myself. So quite often, if you have a discussion with a funder, so typically on the funding website there'll be some contact information, usually a phone number. If you have a discussion with a funder, you can put a different kind of project to them. So they won't fund, obviously, animal research, but they may fund a different study. So as Sarah was mentioning, there's many different kinds of clinical research that you can do, including natural history studies, which show you more about the background of the disease, so how the disease progresses in patients over time. And those kinds of studies would perhaps be more easily able to get funding from those funders. So I think really you just have a discussion with the funder. So typically, in my experience, I found that funders are quite open to having discussions. So if you have a couple of ideas that you want to put to them, if you phone them up and talk through the ideas, then typically they're quite happy to do that. And usually they'll actually help you change your application so it better suits what they are actually looking to fund. So I think, yeah, that's my advice really, is to just have an open discussion with them and perhaps put together a different proposal that's better suited to what they want to fund.
Brilliant. And on that point of natural history studies, Sarah, a question for you. How valuable do you think it is to have a patient registry in place before starting the research project, or the research process, sorry?
Good question. I think in general, a registry is extremely valuable, and in my personal opinion, the patient group is probably the best set of people to do this, because you can have control of what you of the types of information you're collecting, and you can decide who has access to it, and to make sure that the right people, such as sponsors, doctors, etc., have access to the right bits of information, and people can be well connected in terms of finding trials and taking part in trials. So I think it's a great thing to have, but I also understand it's not always feasible to have the resource, say, to sort of get a registry up and running. But ultimately, just making sure you're as open as possible to the idea of collaboration or partnership with a sponsor, because ultimately they have the resource and the funding and the ability to make those connections. So I think a registry is very important, but I wouldn't worry too much about having a registry in place to start those conversations with sponsors, or to start thinking about potentially co-funding or contributing or collaborating on a trial. And in terms of natural history studies, I would say they are important. They're important to find biomarkers, they're important to find endpoints for trials to happen. And so that could be a place, as Oliver's saying, about different types of studies you could support. You could support a natural history study in that way, potentially not needing a registry.
Brilliant, thank you. And you mentioned resources, which is a very good segue to the other question that's been asked. With research being quite expensive to undertake, what would be your thoughts on ensuring the best bang for the buck? So I'll go to Oliver first and then to Sarah.
That's kind of a difficult question to answer, I think. Essentially, you need to look at the disease that you're studying and see what's going to be the most important thing. So, for instance, in alkaptonuria, there was already a drug out there that was licensed for another disease that looked like it could be useful for AKU. So for us, the really big priority there was testing that drug in a model of AKU to see if it had any effect whatsoever. And then if it did, we could then move into doing clinical research. If you're looking at another disease, it could be that perhaps you don't even know what the target is. So maybe your research will be looking exactly at where in the body is the disease having an effect. So is it affecting just the liver, or is it affecting the liver and the kidneys and the lungs, or what area you're actually needing to target? So I think it really depends on the disease. That's probably not going to be a question that maybe patient groups can answer easily. So I think really the first option is to start off by finding the doctors or the researchers that are already interested in the disease. And I think Sarah mentioned in an earlier answer about looking on PubMed to find out who are the people that are looking at your disease. And I would echo that I think PubMed is a great resource for, basically PubMed lists every paper that's ever been published scientifically. And so if you go onto PubMed, in the search bar, you can just type in your disease name and then find probably a couple of hundred papers listing everything that's ever been looked at in your disease. And if you start to see sort of the same names over and over in the papers, those are the people that I would suggest you contact and ask them, what do you think is going to be the best research to fund to have the best bang for your money, as you said.
Sarah, anything to add?
I think Oliver's pretty much covered it all with a very comprehensive answer. I think the only thing I would add is the additional layer that you could bring to that conversation with the researchers that you find on PubMed is just understanding from your perspective, so as a patient group, what are the big unmet needs and what is it that your families really want to look into. It may not always be the most obvious thing. What are the most impactful symptoms and what do you think is really not being managed well? And then you can bring that to the table, to the conversation to see, okay, first then it might not be something that's easy to be funded or researched, but it's also giving the researchers an idea of where you might want to go in the future.
Great, yes. And to add to that, just 2 points. There's a process called the James Lind Alliance priority setting process, which does exactly what Sarah was just saying, which brings together key patients and clinicians and does a very comprehensive survey process to understand what are key unmet needs, which can then help you decide how to prioritize the funding that you have available. That process is quite expensive, but is very useful if you do have some funding in place to try and support the best research possible. There's another useful resource. It's a free ebook that was published. I believe it's called Impatient Patient. And that is a process of how you strategically prioritize the gaps in the research available for your condition to make sure that you speed up the process as much as possible. We've got one final question that's come through. We still have plenty of time if there are any burning questions you'd like to ask. This is aimed at Sarah. And the question is, at what stage should patients be involved in clinical research? I imagine this means patient groups. And how do you start talking to pharma about that?
As soon as possible is the real answer. So as early as phase 1, even before that, in the stage of if you know that a sponsor is going into an area, are you able to contact them then and really get that conversation going right from the start? And I think in terms of how to kind of go about that conversation, it's almost to, just pick up the phone, email, find as many contacts as you can. I think the issue with the pharmaceutical industry is that people really want to talk to patient groups. People really do want to speak out, but there isn't, because it is so highly regulated and so much can go wrong. And there are so many rules around communicating with patients in terms of promotion. Companies are scared. They're scared to reach out themselves, or they're scared also of kind of patient groups saying no almost. So if you're going to be making that first introduction, it may not be the easiest thing to kind of find the right person or get there, but it's mostly to do with the fear of collaborating because of all of the regulation around reaching out to patient groups. So I would just advise to email, as I said, the patient advocacy lead, or even the chief medical officer. There's usually somebody who is in charge of partnerships and outreach, and get the conversation going as soon as you understand or know that they have an interest in the condition that your patient group is supporting, and get the conversation going from there. And the ways that you could collaborate from that early stage are even just to have input into the trial protocol. So these things are usually designed, as I said, with the doctor in mind, or with the help of the doctor, thinking about, how often you might need to go to hospital, which trial sites, what type of tests need to be done. And sometimes they might be a little bit too much, perhaps, for the people actually taking part. And at that point, you can have the input there to help the researchers understand what's feasible, what's appropriate in terms of input into the trial protocol. You could help them to plan the research better, to help find the people to take part, and to understand what's really important to be studying. So the earlier the better, in my opinion.
Brilliant. I don't think we received any other questions, so unless there's something someone really desperately wants to ask, I will say a massive thank you to our lovely speakers. Thank you to Sarah, and thank you to Oliver for stepping in so last minute. Again, want to reinstate best wishes to Hannah to recover as soon as possible. I'd like to thank you for listening and for your very insightful questions. And I do hope that you will join us in a future webinar or at a future event. I would recommend maybe checking out Find a Cure's portal, which is portal.findacure.org.uk, as there are a lot of additional resources on there about medical research, clinical trials, working with academia, working with pharma. So I don't think we've had any other questions, so I will thank everyone again for joining, and I hope to see you at another event. Thank you very much.